Okay, hello everyone and welcome to chapter 16 part 2, Mendelian inheritance. That is the standard pre-recording introduction. Anyways, we are now at part 2. We have covered part 1 and part 4 via pre-recording. If you have not done that yet, I want to remind you to do so because by doing the quiz especially, this is how your attendance is taken. And if my superiors ask me, say, why is so few people doing the quiz and stuff like that, please report it to your parents. I would have to do so. So I will not want to do that. It's more work for me. It's a lot of stupid work for you because you have to talk to your parents about it. It's not fun, okay? So do the assignment by not too late, by the date due, which has already passed. Anyways, we did part 1 and part 4, which is a bit disjointed from these parts. Part 2, and so we're doing part 2 and part 3 online, and part 2 actually has monohybrid cross, dihybrid cross, which I have heard you have already done in IG, so I can go through quite quickly. What new is to you is test cross, and of course maybe you don't remember already, then you have to recap lor. Okay, your foundation is very important in this chapter, and that's why I'm doing it live, and we're going to do a lot of practice together, how to draw Punnett squares, how to do the genetic diagrams, how to do the formatting. Why? Because genetic diagrams in A2 is 6 marks. It can be 6 marks. 6 marks is a lot of marks, and it's very easy to get as long as you follow some specific rules and formatting. So we're going to do a lot of drawing. Okay, next class, okay, I will finish whatever I cannot finish this class, and we'll also go to a stats test called the chi-square test. Don't worry, statistics in bio is crazy easy. Why? Because they provide formula for you, they draw a table for you, all the maths is pretty much done for you. You just need to know how to analyze the results, and we will get there, don't worry. And then the next part, it gets more complicated. Okay, the next part, part 3, is more patterns of inheritance, which is more complicated than this particular part. That's why, again, your foundation is very important. If you don't understand it, if you're lost starting today, you have to ask me questions. If not, the next class, you're going to be very confused. And then when you do past years, you're going to be even more confused. Yay, don't want that to happen. So again, foundation is very important. We're going to draw a lot of stuff. But talking about foundations, we need to start with a few important terms. Now we've done some important terms when it came to part 1 meiosis. This is in addition to that. Okay, pay attention to the important terms, especially those highlighted in red, bold, and then I highlight and like underline, and that's because it comes out and you need and the definition comes out. They will ask you to define this or state what is meant by this, and you need to say precisely what it is law. Okay, moving on. So important terms when it comes to this chapter is 2 things. Number 1, genotype. Genotype is alleles possessed by organism. I'm trying to pronounce alleles right. It's allele, right? Yes. Alleles possessed by organism. This means the genes that you have, right? And phenotype. Phenotype is different from genotype. Phenotype is the observable characteristics of an organism. So what can be seen? So I may have the brown hair allele. For example, I have black hair. Black hair allele, and that would be my genotype. My phenotype is that I have black hair. I can see the black hair, right? It's an appearance. So genotype and phenotype. Phenotype can be influenced by genotype or environment. So I have the black hair allele. This makes me have black hair, but I can also go and dye my hair blonde, for example. And the environment is causing my hair to change color. Okay, just an example. Okay, we're not going to talk about environment that much. This particular chapter has a lot to do with genetics. So we're just going to throw the environment out the window for now. Whatever can happen to the organism, just talk about genes. So what about genotype? Genotype, we have specific formatting. Also, this is the definition that you should have committed to memory already. Gene is a specific DNA sequence that codes for polypeptide. It is represented by the same alphabet. So this is big B and small b, same alphabet still, just one capital, one small, right? It's talking about the same gene but different allele. Okay, I will never say that, hey, this gene has two alleles, A and B. Oh, that is wrong, right? It always has to be the same letter, same alphabet, but it could be capital or smaller. Okay. Gene short form name is usually in italics, and this will become very helpful in your past year questions because it tends to be a little bit confusing. Gene short form name, like HBB, it's italic, means this is a gene name. But usually the gene full name and protein is not italicized. Okay, I think this one, when we do practice questions together, you'll realize it's helpful. Right, what else about genotype? Now, each gene would have two alleles, right? Especially if it's in a diploid organism, you would expect the diploid organism to have two alleles of the same gene. The dominant one is always big B. The recessive one is always small letter. I'm sorry, it doesn't need to be B. It could be other alphabets, but dominant one is always capital, recessive one is small letter. You already know this. I just want to make sure you have your foundations right, so bear with me. No pun intended. Dominant allele always placed in front, and recessive is always placed behind. So what does that mean? Means if in a person who has big B and small b, you have to write it with the big B in front. You can never write it small b in front and then big B behind. This is wrong. Okay, must write like this. Okay. Right, next. Now, what are some other important terms? Right, again, in diploid organisms, in diploid cells, you expect two sets of chromosomes and therefore two alleles for each gene. Now, you could be homozygous or heterozygous for the gene. So homozygous means you have two identical alleles of the gene. You can be homozygous dominant for the gene or homozygous recessive for the gene. So homozygous dominant is big B, big B. Homozygous recessive is small b, small b. Okay, how about heterozygous? Heterozygous means you have two different alleles of the gene. We can call people or individuals with heterozygous, that are heterozygous for a gene, heterozygotes. That's a pun. Never mind. So heterozygotes have two different genes for alleles. Two different alleles for a gene. Sorry, I mixed it up. So two different alleles for a gene. Big B, small b. Again, never small b, big B like that. The dominant one is always in front. Now, why am I going through this bit by bit? It's because they will ask. Okay. What is dominant allele? You already know this, you know it, but how do you put it in words? You know that, oh, if it's there, it will get expressed. But how do you put it in words? Let's go. Number one, it's expressed in the phenotype, even when only one copy of the allele is present. Okay, so it's expressed in heterozygotes and homozygote for the allele. So either heterozygote or heterozygous dominant. So this or this will have the phenotype that's dominant. Okay, so if you're heterozygote, I repeat again, or homozygous dominant for the gene, you would have a dominant phenotype. See, phenotype is the appearance. Now, how about recessive? You know what it is, how to say it. It is expressed in the phenotype only when two copies of the allele are present. In other words, it's only expressed in a homozygote for the allele, or only expressed in homozygous recessive, if you are homozygous recessive for the gene. So small b, small b. I hope you don't have any questions so far, because these are stuff you should already know. Okay, just I'm giving you the words to describe it. Alright, so let's do a little example here. I did say black hair just now, but let's assume this is not a real example, but this is just, you know, an example. So big B, it could be allele for brown hair. Small b could be allele for blonde hair. Now, honestly, you can use many different alphabets for this. You can assign, especially if there's no specific way to express it. Okay, that means they didn't tell you what exactly to use, or you didn't learn how exactly to put it. You can use whatever you want. Really, you can use big R, small r. You can use big H, small h. Now, I want to recommend again certain alphabets, I'm a bit biased towards certain alphabets. For example, big C, small c, the capital and the small letter looks about the same. So it's quite confusing if you write really fast. You don't know whether it's big or small, okay? So that might be a problem. I also don't recommend you using things like big P, small p. Even though you see in my slides, it's actually not a great idea. I found out later on. So recommend you using the alphabets that look different when it's big or small. Anyways, let's do this. So let's again break down what the genotype and phenotype could be if the alleles are like this. So homozygous dominant phenotype would be brown hair. Okay, if the person has big B, small b, the person is heterozygous for this gene, and this person would have no response. I'm gonna wait 2 seconds. Seriously, guys?
Brown hair.
Hey, thank you for that. So brown hair, and then if you have small b, the person is homozygous recessive for the gene, and the person would have blonde hair. So this here we call a recessive phenotype. The brown hair we call it a dominant phenotype. Okay, I just want to make sure you have that terminology very clear. But I also want you to know that sometimes symbols can be written differently. Sometimes, like sickle cell anemia, you have particular terminology that you have to use. In sickle cell anemia, HbA is normal allele, HbS is sickle allele. It's like this. You cannot use big A, small a. Cannot. You must use this for sickle cell anemia. Okay. There are also genes that can look like this. Okay, instead of having, because this gene is on the X chromosome. Okay, so sex chromosomes one work differently. We'll get into that next class. Right? You can have normal little plus there. It could be normal dominant allele. So it could be X minus, could be a recessive allele. Shucks, I keep saying it wrong. Never mind, you know what I mean. For example, also like ABO gene for blood root. This is specific. Also, this is specific. This is specific. You can have big I and then small little a at the side, big I and small little b at the side, and then small I. What does that mean? Well, we'll cover that later on. But I'm just telling you that, hey, it could be written different way. It could be even written like this. It depends. If they give you in the question, you should use it. If you learned it and I tell you it's specific, like HbA and HbS and Ia, Ib, and I, right? These are specific. This means you need to remember. For now, don't worry yet. I just want you to be prepared and do not panic when you see them. But I will go into the details later on. Okay, if you have any questions for terminology, you should put them in the chat about right now while I take a sip of water. No? Don't have? Okay, let's go. Okay, so Mendelian genetics is named after this guy called Mendel, which is the father of modern genetics. And he actually didn't know about genes and chromosomes because he was around in like the 18 something, right? And he's not even a really he's a scientist of his sort, but his main job is actually being a monk. And I guess being a monk and maybe a little bit away from society, he had a lot of time. I'm just making assumptions about his life right now. And he planted a lot of plants and basically studied the inheritance in pea plants. And again, he didn't even know about chromosome and genes yet, but he completely got the points of inheritance. He completely understood it, you know. So whatever we know about classic genetics so far, you know about classic Mendelian genetics so far, the dominant recessive thing, this is all his work. Okay, so he did it in mostly pea plants, by the way. So you will see a lot of pea examples, PEA, no laugh, don't be immature, right? Yellow, green pea pods, round crickled seeds, tall sharp plants, purple, white flowers. He did it mostly in peas. That's why the examples we use are mostly peas as well. Okay. He did 2 main experiments, which we're going to learn, and I think you already have, but we're going to go and do it again because you probably have forgotten. And again, foundations are important. Right, 2 main experiments. Number 1 is a monohybrid cross, and number 2 is a dihybrid cross. Okay. In monohybrid cross, we deal with one gene, one trait, one gene. Dihybrid cross, we deal with 2 genes. And we're going to cover both today. Okay, so what did he do in monohybrid cross? He basically took 2 purebred plants. Okay, purebred means he took the tall plant that has always historically been tall. Means he breeds tall with tall, he gets tall plants. It's always been tall. Right? Purebred short pea plant means you take the short and short, you breed together. It's always short. You never mix it with the tall plant. So both are purebred, and actually what it is that both are actually homozygous for the trait. So happen. And he takes these purebred plants and calls it the parental generation, and he breeds it together and realized that all the offspring are tall. The offspring we call the F1 generation. Now F1 stands for filial generation, not something you need to write an exam, but for you to know for fun. So when he took this F1 generation and then he crossed it with itself, okay, actually maybe he took the flower and then he just 2 flowers and he just like rubbed it against each other, the pollen will exchange, right? So it's self-fertilizing, and realized that all the offspring, again, plants, right, don't give birth, right? So you rub the 2 flowers together, then you wait for it to have fruits, then you take the seeds, then you go seeds, you take a plant, and the seeds, which is the offspring, will become the plant. And the plants is the F2 generation. In the resulting F2 generation, he obtained this amount of short pea plants, okay, and this amount of tall plants, and it's around a 3:1 ratio. Yay. So this is the story, but this is not genetics yet. So our job is to show it using a genetic diagram. So as we talk about it, I'm going to show you what we need, and then I'm going to make you draw a few on your paper. Okay, so let me do this first. Every genetic diagram must have, number 1, parental phenotypes. So what happens in the first generation? What happens in the parental generation, the first generation? Purebred breed with purebred. Purebred tall breed with purebred short. You don't need. You have to not forget the X. X means to cross. So tall cross with short. The X is important. Well, minus mark if you don't put it in. Number 2, parental genotypes. Okay, phenotypes again is the appearance. Genotypes is the genetics. So tall purebred is big T, short is small t. The gametes, okay, right? One thing I always tell people is, look, there are 2 different things here, 2 alleles, right? In gametes, it's by a process of meiosis, right? So each cell would have 1 chromosome, 1 allele, right? So 2 allele become 1 allele per cell. 2 allele become 1 allele per cell. This is the process of meiosis that's happening. Okay. Now, actually, you don't need to write 2 because it's the same type. You can just write 1 because it's the same type. Right? You can just write that and that. And that's fine. Now, the next thing you need to write is the offspring genotype and phenotype. This should be in a Punnett square. You must draw a Punnett square. This is a very easy example. We will still draw the Punnett square, and you must circle all the gametes. So you can realize that this is circle, this circle, this circle, showing that they are in distinct different cells. The gametes are different cells, right? Circling it. So Punnett square, big T, small t put together show the phenotype. This is a tall plant. And the last but not least, you have to show the probability or ratio. Now this depends on the question, but we should, even if they don't ask, we write a few general statements describing our data. So this data shows that, hey, all plants are tall. The probability of getting a tall plant is 1, which is all the time. You will get a tall plant all the time. So these are the 5 things you need for genetic diagrams. Again, must draw Punnett square, must circle gametes, don't forget the X. Now what we're going to do is, I'm not going to talk about a second, this is the filial one, F1 generation. I'm not going to talk about F2 yet, but what I want you to do is to take a paper, because we're going to do quite a few practices together. I need you to write this at the side. Okay, make sure there's enough space for the offspring genotype and phenotype here. Okay, maybe write like 7 of these. Okay, 7. This is the highlighter. 7 times, please. On the left-hand side of the paper, leave the right-hand side blank so you can draw. Again, remember to leave a lot of space here so that you can draw Punnett square. It gets very complicated during a dihybrid cross. Okay, maybe you will need to spread this across, I don't know, 3 or 4 pieces of paper. I'm serious. This is why I had it live, because I want you to practice. You don't practice this, you get it wrong during the real exam. I have students who like, yeah, I remember this. Turns out in the exam, they don't remember it precisely. They will miss out small things like the cross. Like, they're like, so easy, why don't you draw Punnett square? Draw the Punnett square. There's marks for it. It's 6 marks. Okay, it's free. Do it. So you're gonna draw it 7 times today. More next time. So 7 times today, 3 or 4 papers. Spread it across 3 or 4 papers. Leave enough space, please. You will not regret the space. Okay, I'm going to stop talking now so you have time to draw. Draw nice, so that you can keep as notes. Okay. When you're done, type in the chat done so I know, and I know when to move on. When you draw 7 times, you can include the first one, which is this monohybrid cross, the first generation. Title for those things, monohybrid cross, the first, and then copy back the first one. Let me know if you're done. Sorry about that. Yay, one person says done. Again, if you're done, type in the chat, and not I'll wait for nothing. No waste time. Also, if you see me stretching, I just feel I feel I have age in front of the computer screen. Neck hurts. Okay, liking Beatrice punya post helps too. You can just like, yeah, that's fine. Done. Everyone else? Please have done. Waiting for more people to respond. So 10 people done so far. How about the other like 21? 12 people done so far. Thirteen. Sorry. Great. Okay, I'm going to give you 1 more minute. I'm going to start. Remember to draw the first genetic diagram. Okay, this one that you see on the screen. The first genetic diagram, the monohybrid cross. Alrighty. So, yep. Yeah, I said that from the beginning. Chap. Okay, so we have the first cross here, telling us what happens when we take two purebred one purebred tall and one purebred short plant, cross it together, we realize that the F1 generation are all tall. Now we take the F1 generation and we self-fertilize. So here you can put a little bracket beside your parental phenotypes and just write F1 phenotype. All right, just write parental phenotype bracket F1 to let yourself know that this is the filial 1 generation. So you are self-fertilizing, so tall. The genotype is big T, small t. They are heterozygous. Okay, when we talk about F1, you need to be very clear that F1 is heterozygous crossed with heterozygous. Okay? Heterozygous crossed with heterozygous. Parental is sort of like general. But when we say F1, F2, we are very, very specific. We're talking about this procedure. So you should write this in the second diagram. So what's next? Gametes. So gametes, you have big T, small t for both plants. And then of course, after this, you're gonna write the F2 genotypes and phenotype. Again, on your paper, it should be offspring, and you can bracket F2 just to let yourself know that it's F2 is very specific cross. Draw a Punnett square. Now, of course, in the exam, you need to draw all the lines. It has to be very neat and tidy, but this is your notes. Please draw the lines later, just draw roughly for now. It can be defined later if we have time. I'm gonna move on. So gametes, you're gonna put it in the table. Okay, so this fella put here, that fella put there. Actually, it doesn't matter which fella you put where. It's the same as long as you put one organism, one row, one organism, one column. It's fine. And you realize that this is big T, this is big T, small t, and they're all tall. The last one is small t, which is short. So the probability ratio is 3 to 1. Ratio of tall to short plants is 3 to 1. Probability of getting a tall plant is 3 over 4. 4 is actually 3 plus 1, okay? And probability of getting a short plant is 1 over 4. And that really explains the basis of the entire monohybrid cross, genetic basis. So again, make sure you have a few things in your diagram as you're drawing it right now. I'm going to give you some time. Make sure you have the cross. Make sure that you have the Punnett square. Make sure that you have circled your gametes. All of the gametes. So this is monohybrid cross. As you can see, is one gene, two different alleles. Yeah. And soon we'll do two genes. But before that, I'm going to introduce something called test cross to you, which I'm still waiting because I know you're drawing right now. If someone could tell me if they're done, that would be great. Then I can move on. What Michael says is done. Okay, great. So moving on. This is the monohybrid cross done. It's revision for you, right? It's not new to you. Before we go on to dihybrid, we're going to do a few things. We're going to ask this question. Hey, why didn't he find the exactly 3 to 1 for everything? Now, it's important to know here that we're talking about probability. And you have learned this in maths, right? All of you take maths. Most of you take maths, right? And that probability only applies on the individual. Okay, just because there's 50% chance, for example, of getting a female or a male, doesn't mean that all families have equal amounts of males and females, right? Okay, so the same thing applies here, right? Just because the probability is 3/4, or the ratio is 3 to 1, it doesn't mean that the ratio of the numbers in the second generation, F2, will be exactly that ratio. The more examples, the more samples you take, the closer it will go to that ratio, but also not exactly. And that's because the probability is applied to every single plant. For every individual plant, there is a certain probability of being tall, certain probability of being short. Okay, I hope that makes sense probability-wise. Okay, so don't be confused about that when we talk about probability. Now, before we go to the hybrid, I mentioned that we're going to do test cross. What in the world is a test cross? Now, this is the situation here. Put the monohybrid cross you thought about aside for a while. You're randomly walking in Mendel's pea farm of some sort, but you randomly found a tall pea plant. How do you determine if its genotype is purebred, means homozygous dominant, or it's heterozygous? We use the test cross. Okay, test cross is a method to determine the exact genotype of the dominant or phenotype of a dominant phenotype organism. So in this case, we are using the tall short example. So if it's tall, is it again big T or small t small t? Big T small t. What is the solution here? In a test cross, we cross the individual with an individual homozygous recessive for the gene. AKA, in this case, we cross that tall pea plant that you don't know what its genotype is with a short plant. The short plant will always have the genotype you expect, right? Because it's recessive. Short plant is definitely homozygous recessive. It cannot be anything else because it's a recessive phenotype. So it must be homozygous recessive, right? So we can cross this tall pea plant of unknown genotype with this short plant. We can observe the offspring and figure out whether it's homozygous dominant or heterozygous. Okay, this one you don't need to draw a diagram yet. We will draw a diagram later. Okay, let's just pay attention here. Okay, if the tall plant is homozygous dominant, so big T, okay, for example, you don't know this, but if it is, imagine big T crossed with small t, you get what? So you draw a very rough diagram, a Punnett square here, didn't draw the whole thing. You realize that all offspring will be tall, right? Because purebred cross with purebred. Homozygous dominant cross with homozygous recessive. All will be tall. If the tall plant is heterozygous, on the other hand, heterozygous cross with homozygous recessive. Now this is different. So the gametes will be this and this, right? So the offspring can be tall or short. It doesn't all have to be tall. And of course, you don't do it in one cross. You do it many times to make sure that, what do you call? that to verify your results is not just by chance. Okay, do enough sample size, make sure your experiment is reliable and you can reduce anomalies, right? So after that, you look at the offspring. Is it all tall? If it's all tall, it's homozygous dominant. If there is some short, just even one short among that, like, you know, 10 tall, you know that, hey, this tall plant must be heterozygous. Understand? Doesn't even need to go to the 1-to-1 ratio. As long as it has a short plant as the offspring, you go like, this one heterozygous. Because if it's homozygous dominant, it has no chance of it becoming short. It must be tall. So that's a test cross. Again, if you have any questions, put them in the chat. And what we're going to do now is we're going to do, ignore that first, we're going to do an example question. Yay, look, dogs, cute. This is a Dalmatian. They have 2 alleles. One is a black spot allele, which is dominant. Brown spot allele is, I'm saying allele is recessive. The breeder wants to know the exact genotype of a black spot dog. Black spot dog is dominant phenotype, right? It tells you that this fella is dominant. So big B, black. Small b, brown. You can write allele. Right? So she crossed her dog with another brown dog, and puppies are produced and showed all black spots, and the same results were produced. So what is the dog's genotype? With this information, you should already be able to predict. Don't tell me the answer, because you are going to draw a genetic diagram to show it, and we are drawing it together. Actually. So I'm going to do mine in the OneNote here so that you have something to refer to. It is under A2 Doodles, Chapter 6, Mendelian Inheritance. Just like that. The example question is here. I know you didn't ask me, but you may in the future ask me that whether that let big B equals to black allele thing is compulsory, and the answer is no, it's not, but it does help me be less confused when you know you suddenly forget, then you got something to refer to lor. I don't like red color now. Let's use blue. Alright, let me know when you're done so we can discuss. I'm hungry. Half an hour more. Let's go. If you're done, let me know. If you're done, let me know. All right, so I think this one's quite easy, but you know, practice makes perfect, right? So this is black spot because all the puppies showed black spots. So your answer for the genotype should be, hey, if all of them show black spots, which is a dominant phenotype, this means it's probably big B, big B. So what you need to do is to just explain that by writing down big B crossed with the homozygous recessive. This is what we mean by a test cross. The gametes, don't forget to put a cross here, don't forget to circle the gametes. Again, since there's only one type of gamete, you don't really need to draw it twice. Okay, draw the Punnett square. Okay, I'm going to draw a horrible one. You can draw a nice one with ruler, because you know, mine very hard to do ruler. Big B, small b would give you all, oh sorry, not tall, all black spot. That is the genotype and phenotype. Make sure all the lines are intact and pretty, not like mine. Mine's just awful, huh? Okay, I give up. I'll fix it later if I have time. So the probability is, okay, again, they didn't ask, but usually in exam, they will ask very specific questions. But now we're just gonna briefly describe our data, which is all dogs are black or have black spots. And this proves your genotype right here. Yay, well done. So, yay. So that is monohybrid cross, that is test cross. Let's go back to the previous slide here. Let's lay down some important terms we've learned so far. Number one, what is purebred? Purebred always produce offspring with the same genotype. Okay, if you cross them together, these are homozygous individuals. So in exam, when they say, oh, this is purebred, they are homozygous, confirm plus Chap. Okay. Parental generation is a general term. Actually, it just means initial generation. Okay, anything can be parental as long as they are the first cross. But when we say F1 general, this is specific. When we say F2, this is also specific. F1 is the result of crossing two purebreds, must be pure, and all of F1 offspring, okay, all F1 offspring as in F1 generation, okay, if you're confused, you can change this word to generation, are heterozygotes. It's confirmed. So if they say, I crossed two F1 individuals, this means crossing two heterozygotes. F2, also specific, crossing of two heterozygotes punya result. Okay, this result can differ for monohybrid. Okay, only for monohybrid is 3:1. Different for different patterns of inheritance. F2 generation will differ, but it's a result of F1, two heterozygotes crossing together. Okay. All right, and last but not least, we have learned that test cross is a method to determine the exact genotype, and essentially, it's just take the random thing you found, take the individual you don't know the genotype of, you cross it with an individual that is homozygous recessive for that trait. So these are the important terms we learned so far. Now with this settled, we can move on to dihybrid, which you already know, but which means I can just go faster, really. But I'm going to go through it just in case you don't remember anyway. Now, as I said, monohybrid inheritance, we're talking about one gene, one trait, either tall or short. But dihybrid inheritance, we're talking about the crosses of two genes at once. Now we're going to add purple and white flowers into the mix as well. Just so you know, when we talk about all these crosses, all these genes are on different chromosomes, and this matters. This really matters, because this means that there are independent assortment that occurs. Okay, different chromosomes means they can flip or not flip, right? And different chromosomes were independently arranged regardless of other chromosome. That is what independent assortment is about, right? And that matters because this means the combinations are endless. I will talk about that later on again. But the idea is there's two genes, two alleles code for different traits. So here are two of them. So we can do the cross that Mendel did again, right? This time with a different set of stuff. So we're going to cross tall purple flowers. This should be your fourth genetic diagram. Okay, this is dihybrid cross. Label it nicely so that you know what you're getting yourself into. You know what to, I mean, when you refer back, at least you know what it is about, okay? So this is a dihybrid cross. Label it. This is your fourth genetic diagram, crossing tall purple flowers and short white flowers. The parental genotype is big T, big T, big P, big P. These are purebred, tall purple. These are purebred, short and white. So small t, small t, small p, small p. This is the dihybrid cross. This is Mendel's experiment. Okay. I hope you are writing as I'm talking, just saying. Okay, so realize that these are 4 chromosomes, right? 4 different chromosomes, 4 different alleles, 4 alleles. Okay, 2 genes, 4 alleles, because 2 alleles each, right? So this is a diploid number, actually. It's a diploid, so-called. This is a diploid organism. After meiosis, it should be haploid, which means the gametes would have 2 letters in them. Diploid got 4. Divide 2 is 2. So the resulting gamete should have 2 alphabets in them now. Okay, 1 for each gene. So big T, big P, small t, small p. Make sure it's circled because it's gamete. Make sure there's S, X because it's cross. Next one is the F1 genotypes and phenotypes. Okay, again, very, very simple diagram, but you still need to draw the Punnett square. It will result in tall purple flowers, and the genotype should be big T, small t, big P, small p. Now make sure that the T is grouped together, the P is grouped together, and the dominant one is in front first. Okay, you cannot write this. You cannot write this. You cannot write anything else except this. Must we write the phenotype below the genotype in the Punnett square also? Well, ideally, yes. You can write it outside, but you need to link it. Means you must, you can write it out of the diagram, but so you write this F1 genotypes, and this one F1 phenotypes can. But can you imagine if you have like a huge diagram and a very complicated Punnett square? There's going to be a very, very long list of F1 phenotypes linked to its genotype. So might as well you just write in the table lah. Write in the table, write in the table. So the answer is yes, you can, but you should not. Alright, of course, the last is the probability and ratio. All plants are tall and have purple flowers. The probability of tall and purple plants is 1. Not tall and purple plants, sorry. Tall plants which have purple flowers is 1. Well, in the exam, the phrasing will matter more. Now, this is how to obtain the F1 generation. Now, F2 generation is where things go completely insane, right? And I think you're probably expecting this already. So F1 crossed with F1 means heterozygous for both genes, crossed with heterozygous for both genes. And the gametes, okay, would be many, many combinations. Why? Independent assortment. Can arrange in whatever thing they want. So you can have all these combinations. It's possible. Now I want you to stop writing for a while for this F1 thing, because we have tricks up our sleeves. I have a trick to teach you to make your life easier, and I want you to follow this trick because it saves me a lot of marking pain and also saves you a lot of errors. Okay, save you from a lot of errors. So just so you know, Oswald asked though, is it possible to have more than one type of gametes for some genotype? And he showed this. Well, this is not relevant at the moment because we are specifically talking about crossing the F1 together. And for that, it is possible. Yes, it is possible. I'm going to show you how to get the different combinations of gametes now. First, you take the first one, right? You take the first one, we're like, okay, which one can I pair it with? It could be big P, big T, big P, or it could be big T, small p. Okay, again, CP is a very stupid, like it's a bad letter to choose from, but I'm just gonna stick with it because it's in the slides already. Alright, the next letter you choose, I'm gonna change color so that you know, can see better. Blue color. Right, so the next letter, small t, right, can pair with what? Can pair big P and can pair with small p. So big small t, big P, small t, small p. So you have 4. Now for this, okay, although it's not relevant right now, of course you can have different gametes. Right, these two are the same, so there's only one type in front, so it could be like this, or it could be like this. So big T, big P, or big T, small p. You don't have 4 because this is the same line essentially, because this is 2 of the same, alright, but there's still types. Okay, that's how you determine them. But I don't want to see in exam, I don't want to see these lines. No one wants to see those lines. We just want to see the gametes that circled together with the arrows. This is what we want to see. We don't want to see these. No one. Okay. SPM does that a lot. I don't know if IGs, your teachers used to teach that to you, but we don't want to see it during A-levels. Okay. We are way past that. We are better than that. So you should have all these 4 combinations. Now I told you don't write yet because I have a trick. Okay. Right. And here's a trick. It's a huge table. It's very complicated, right? And it's a pain because you need to look at it very carefully. You must make sure you do the right thing and It's a bad idea. So I mean, it's a nightmare. So, but if you arrange with the dominant gametes first, it makes life easier because we have a trick. Okay, you must arrange your gametes in a certain method in order for the trick to work. Always arrange with your dominant gametes first. So big T, big P comes first, then big T, small p comes next, then the dominant behind, then the recessive behind. Okay, always the dominant first. Okay, this one small t, but dominant first, and then the recessive at the end. Same for this. Same when in the Punnett square. So this is the dominant first, and then followed by that dominant, and then something else, dominant. So most dominant one first. Get it? Must be in this order for the trick to work. Now I'm going to draw the tricks in the OneNote so you can keep it. As notes or refer later on. Okay, so now with this, the normal person will go like, okay, let's start. Big T, big T, big P, big P. This one big T, and this one big T. Okay, small, big P, and small p. This is the average person. Maybe you, may not be you, depending on how smart you are. So what years of experience and struggling with this has trained me to do is to find another way. Right, this way might look a little bit more messy, but would save your brains. So what do? We do it row by row first. So big T, big P, we know that dominant will be in front. So we're going to write a big T, leave a space, write a big P, leave a space. Okay, then we're going to do it for rest. All right, make sure you leave some space at the bottom area for your phenotype later. Okay, then you write big T, big P, leave a space. Big T, leave a space, big P. Big T, leave a space, big P. Nice. Next, big T is always in front because it's dominant, but small p is always behind because it's recessive. So we're gonna write big T and then leave 2 spaces. Okay, I'm gonna make a lot more space there. Leave 2 spaces, small p. Okay, make sure your small p is distinctly different, so distinctly smaller than the big P, if not it will get very confusing. Okay, or you know what, in the first place, don't choose the alphabet, but I lazy change all my slides now in the middle of a class, so I'm just gonna stick with it. Big T, tiny p. Big T, tiny p. Messy. It is kind of like the words are not very pretty, but it doesn't matter. You're less confused. All right, and next one, you get the drift now. The small t is always at the back because it's recessive. So space, small t. I'm going to write it in the middle of the cell because big P is always in front and something behind. So t, small t, big P. Small t, big P. Don't forget to leave some space for your phenotype later on to write in the same box. All right, next one, you get the drift. Space, small t, space, small p, because the recessive is always behind. Continue for the rest of the blocks. Oh, make sure your small p is tiny, if not you get confused. All right. Then we're going to do it row by row. Okay, I'm going to change color just to make sure you can see the difference. So we're going to take big T and big P and fill in all the blank spaces here. So big T, big P. All the blank spaces. Because all the blank spaces should be in the right position already. Alright, then this one is big T, small p. So big T, small p for everyone. If you have any questions, let me know in the chat. Am I too fast? Of course, look, it'll be a bit ugly because, you know, spacing. It's okay, doesn't matter as long as your answer is correct, man. Again, see, the big P, small p, you need to be quite distinct in order to differentiate them. Make sure they are different. Right, next, small t, big P. Small t, big P. Small t, big P. Small t, big P. Then small t, small p. Small t, small p, small t. And with that, you're probably done. And this actually reduces a lot of errors because if you're manually doing it cell by cell, it's very likely that, it's very likely that you write something wrong because you didn't see it properly or something. Everyone okay? Everyone's probably bored now, huh? Okay, so since no one stopped me, I'm just gonna go and talk a little bit more. So what's the advantage of arranging the gametes a certain way? Now, by arranging it, we can draw some triangles and figure out our phenotype. The phenotypes are in nice triangles. How do we achieve this? Well, I'm going to show you here first. You realize that I can't. Okay, you realize that this and this line here, this triangle here, these are all dominant phenotypes for both. They're all dominant phenotypes. Let me show you. I'm going to use a different color. This is tall and purple. Tall purple flowers, okay? I'm just going to write tall P for now because lazy. Okay, so this tall purple, is this tall purple? Yes, it is. These are all big T, big P, right? So it must be tall purple. This one also, because of the big T, big P here. I know I already say it's triangle, but you realize that I'm still checking because it's important to check your work always. So again, you can see here big P, obviously this tall purple. Yay, very quick. Double check, realize that's correct. Alright, the next triangle, I'm going to use a different color now, is here. It's actually more like 3 points. It's here, here, and here. These three cells, the triangle, has tall, dominant phenotype, but it has two small p's. So this is white, tall, and white. Tall plant, white flowers. Yay. Very nice. Next, triangle. Here, here, and here. So this is short but purple flowers. Short plant, purple flowers. Okay, because it has big P but two small t's here. Again, this will only work if, again, you arrange the gametes properly. If you do not, it's going to turn out differently. The last one is short and white. Okay, I have another nice slide here for you to refer to. It makes more sense. And if you calculate the ratio, you should get a ratio of 9, 3, 3, 1. So 9 purple, top purple, 1, 2, 3, 4, 5, 6, 7, 8, 9, 9. Okay, going to use red color again. Ratio should be 9, 3, 3, 1. This is your F2 ratio for a dihybrid cross. Now, in the exam, is not sufficient maybe to write a ratio like this, especially if they are very particular. So make sure you tell them what the ratio is about. Oh no, what did I do? Go back down, go back down. Right, tell them what the ratio is. Okay, this is tall plant, purple flower, to tall plant, white flower, to short plant, purple flower, to short plant, white flower. Ta-da! And you get a nice 9-3-1 and a diagram with minimal mistakes. Yay! And it's 1 o'clock. So yeah, you get a probability of 9 over 16 for tall purple flower. So 16 is actually 9 plus 3 plus 3 plus 1. So tall purple flower is 9 over 16, short purple 3 over 16, so on so forth. And this is your F2 cross. F2 offspring of your dihybrid cross. And with that, I think our class stops here. Don't worry, we didn't finish. It's okay. I expected this to happen anyway. We're gonna do more when we come back. Maybe one more example, test cross, and then we're gonna move on to chi-squared test next class, which is tomorrow. So yeah, that's it for now. If you have any questions, leave them in the chat. If not, go have lunch. Bye bye.